Good day, everybody. Thanks very much for joining in on this talk, and thanks to the organisers for inviting me to give it. I'm going to give an overview on HIV vaccines and immunotherapy. I've recently moved to the University of the Witwatersrand in Johannesburg, South Africa, but all the work I'm going to be talking about is from the NICD and in collaboration with these partners. So just an outline of the talk. I'm going to touch on COVID since it is dominating our thoughts at the moment, but I'm mainly going to give an update on HIV vaccines and some of the new concepts. I'm going to talk about passive immunizations and discuss the AMP trial, and then talk about the BNAB pipeline. But also throughout the talk, I'm just going to reflect on lessons learned and where we're going. So this slide shows the devastating statistics for the numbers of deaths caused by these two viruses. On the top, you can see that in the first 6 months of this year, we've already lost more people to COVID than we did in the whole of last year. And clearly this is a trajectory that we don't want to continue, but fortunately, as you all know, we have highly efficacious vaccines. And so we hope that this is really going to turn things around, because we certainly wouldn't want that graph to look like the bottom graph of HIV, which, as you can see, has been spanning for 40 years and we've lost 36 million people. And at the peak of the HIV epidemic, actually the number of deaths per year were very similar to that of COVID last year, about 1.8 million people. But that decline that we see is really because of the introduction of antiretroviral therapy. But that decline is not going all the way, and that's because we aren't able to get on top of transmission. And that's because we don't have a vaccine. So we really do need a vaccine to prevent HIV transmission to really get that curve down. So the other thing that's striking about these two graphs is the colors. So as you can see that HIV is primarily a problem in Africa. We've had very few deaths in Africa from COVID, thankfully. But really, I think it does speak to the fact that we are one world, and we really do need to think about these things collectively, particularly for viral infections, which, as you know, spread very efficiently. So just to touch briefly on COVID vaccines, because it has been amazing to watch, especially as an HIV vaccinologist, and we all know that just 2 months after the virus was discovered, there was already a phase 1 trial. And that's really remarkable, and I think something we in the HIV field should take on board. But of course, we know that they didn't start from scratch. In fact, many of the platforms that they're using were in fact developed for HIV and for other pathogens too. And so they were able to rapidly repurpose them for COVID. But there's still hundreds of candidates in preclinical and early clinical development. There are currently 32 efficacy trials, and we have 8 vaccines already approved for use. And that's really in stark contrast to where we are with HIV vaccines, where after 20 years, we've done 7 efficacy trials and we have no approved vaccines. So why has it been so much easier to make a vaccine against SARS-CoV-2 than HIV? Of course, a lot of it, is to do with the virus itself. HIV is a much tougher target. And that's also because HIV is a chronic infection. It integrates into the DNA. Whereas SARS-CoV-2 is an acute infection, the body, the immune response is able to effectively clear the virus. HIV is also a moving target, whereas SARS-CoV-2 is a relatively stable genome, of course less stable than we thought it was in the beginning, but nevertheless certainly more stable than HIV. But a lot of it also has to do with the antibody response. So for SARS-CoV-2, almost everybody develops good cross-neutralizing antibodies, and that's both after infection and vaccination. Of course, in HIV, very few people develop broadly neutralizing antibodies, and they take many years to develop. And that's because these antibodies often need extensive somatic hypermutation to show breadth, whereas there are very few SHM is required for antibodies to SARS-CoV-2. Also, the response itself to SARS-CoV-2 is very focused on a few epitopes, whereas for HIV, there are multiple epitopes on the envelope glycoprotein. So if we just review the vaccine trials that have been done for HIV over the last, more than 20 years before when they started, what you can see is that certainly there's been a lot of activity recently, and that's good, but it's been at a slow pace. And as you can see, there are lots of crosses. There is the one tick, which we all know, the RV144 vaccine, that showed moderate efficacy. And that trial really spurned the Hvtn 702 trial, which is basically to build on the success of RV144. And as you can see, disappointingly, that vaccine failed to show efficacy, and I'll discuss that in a minute. We still have one trial that is in efficacy testing, and we await the results of that, the Hvtn 705. I've also put on this slide, and I'm going to be talking about it later, of course, the 2 AMP trials, where we did get a signal. As I say, I'll talk about that in a minute. So just to mention the Hvtn 702 efficacy trial, which started in October 2016, and it was designed to really build on the success of the canary pox and the GP120 vaccines that were tested in RV144. And it was done in South Africa, over 5,000 high-risk men and women. And the hypothesis of this trial was to test whether non-neutralizing antibodies, particularly those to the V2 region, are potent enough to achieve vaccine efficacy of greater than 50% for at least 2 years. Now, very disappointingly, this trial was stopped in February for futility, and this is the results from the NEJM paper published a few months ago, really just showing that there's no difference between the vaccine and the placebo arm. And of course, that's really got us all scratching our heads. You know, why were we not able to replicate the RV144 signal? But there were a number of differences between these 2 trials, and I've listed them here. The adjuvants were different, the sequences were different because the 702 vaccine was modified to target clade C. There was also more boosting, which we know affects immune dynamics. The viruses, of course, are different in the different regions, and in fact, the clade C sequences are more divergent from the vaccine than in Thailand. There's differences in host genetics and other host factors. And then, of course, we did know about the immunogenicity from the Hvtn 100 trial, which was the earlier trial testing the same clade C version of the RV144, and we know that it did induce lower levels of V2 antibodies. But clearly a lot of work needs to be done here to try and figure out why this vaccine failed to show a signal in South Africa. And then just to mention the one efficacy trial that is still in the field, the Hvtn 705, 706 trials, which use an Ad26 with a mosaic insert, and that's to cover many viral subtypes. It also has a clade C GP140, and again, this vaccine is designed to elicit non-neutralizing antibodies. And the rationale for this vaccine is the work that's been done in non-human primates, where the non-neutralizing antibodies were shown to provide good protection. So the results from this trial are expected in 2022-23. But of course, we really want a vaccine to elicit neutralizing antibodies, and there's been work done over the last 10 years by multiple groups, really showing how that happens in natural infection, how these rare individuals develop these very broadly neutralizing antibody responses. As I mentioned, they take a long time to develop, and people have gone to great lengths to identify the original B cell or the unmutated common ancestor, and then mapped out the co-evolution between the virus and the antibody to really try and figure out what are the critical changes that are required in order to make an antibody broadly neutralizing. And so this has informed quite a lot of the subsequent vaccine approaches that have been developed and that are now in clinical trials. So the one by Bill Sheaf and colleagues, which uses EOD-GT8, which basically homes in on those unmutated common ancestors, or the germline antibodies that give rise to CD4 binding site antibodies. And there's already some encouraging preliminary data to show that indeed in humans you can find these rare B cells, and of course the next step is to boost those to see if we can really get a broad response. There's also fusion peptide priming. Peter Kwong and his team are using a short peptide, and then again boosting to try and mature the response. And so the boosting has really benefited as well from the work that was done many years ago by John Moore and Rogier Sanders in terms of identifying to create a soluble version of the trimer, and showing that indeed these trimeric versions, which are more native-like, can in fact stimulate some neutralizing antibodies, autologous neutralizing antibodies in animals. And again, these trimers are in phase 1 clinical trials in humans. So lots of exciting new concepts, but also different ways of doing the trials. So and this idea of trying to coax the antibody response along a pathway towards breadth, following what happens in natural infection, is also being mimicked in a vaccine, where vaccines that are subtly different are sequentially used in an immunization strategy. And then, of course, also this idea that rather than overloading the immune system, let's give small doses of antigen and build up to get a better immune response. So now I want to turn my attention to talking about passive immunization, which is a faster way to deliver immunity. It's instant immunity, but of course it's not long lasting, the antibodies would need to keep being infused. Of course, this is not a new idea. Convalescent plasma has been used for more than 100 years to treat infectious diseases, but of course with the advent of new technologies, monoclonal antibodies, this field has just exploded. In fact, it's the fastest growing area of in biological products. These antibodies for both infectious and non-infectious diseases. So the idea here is that antibodies are infused before or shortly after infection to provide instant immunity. And the advantages here is that antibodies are highly specific, and so they're less likely to have safety or unexpected side effects. And indeed, we have seen that. The other very appealing thing is that these antibodies can be engineered to increase their functionality, whether it's their breadth or their potency, but also to increase their half-life. And that's been a major advance that is certainly going to help with the use of these antibodies as therapeutic agents. So I now want to tell you about the first clinical trial to show that broadly neutralizing antibodies indeed can work for HIV, and this is the AMP study, which used the VRC01 antibody that targets the CD4 binding site on the HIV envelope. So in this trial, and there were 4,600 high-risk men and women in Africa and the Americas in this trial, they received infusions of VRC01 every 2 months, and they got a total of 10 infusions, so a long-term study of 2 years and real dedication from the participants as well as the study teams in carrying out this, this really incredible study. And then real time, we were monitoring the breakthrough viruses for both VRC01 sensitivity and also measuring the serum levels of VRC01 in these participants using both ELISA and neutralization. So I'm going to cut straight to the result, because overall this trial did not show efficacy. However, when we looked at viruses based on their sensitivity to VRC01, we did see a signal. So these categories were pre-specified before the trial. And so we categorized viruses that were very sensitive to VRC01 at less than 1 microgram per mil. And then we had viruses that were intermediately sensitive, and then viruses that we considered resistant to VRC01, the 3 microgram per mil. And what you can see in the less than 1 microgram per mil group, in fact, the efficacy was 75%. And you can see the numbers there in the placebo arm and in the 2 VRC01 arms. We saw no efficacy in the intermediate or the people who had resistant viruses. And this looks at the data in a slightly different way. So this is now looking at the individual IC80. So this is the amount of VRC01 that it takes to inhibit the virus. So the IC80. And as you can see in the control arm, and this would be the naturally circulating viruses, there's a huge range of sensitivities of viruses to VRC01, from viruses that are very sensitive at the bottom to viruses that are very resistant, although most are in the middle. But that is the range of sensitivities in the control arm of AMP. And as you can see in the 2 active arms, what you'll notice is that there are fewer red dots below the line of 1 microgram per mil, and that's because those are the viruses that were blocked by VRC01. And so that when you take the median of these viruses, indeed the viruses from the 2 active arms had a 2-point-fold greater IC80 than viruses from the placebo arm. So in other words, they were more resistant, which is really what the hypothesis was, to start with. So this is a new piece of data from the AMP study, because of course the other critical piece of information that we can get from the study is what are the levels of VRC01 that are needed to protect from HIV infection. And AMP has been able to give us this answer. So what was done here is that we measured the levels of VRC01 over time and did some predictive modeling to map the PK. And then we took the median midpoint and worked out the inhibitory dilution. And we divided that by the IC80 of the virus, of the circulating viruses. So we know exactly what levels would correspond to the viruses in the circulating viruses in the AMP trial. And so what you can see is that with increasing concentrations of ID80, we see increased efficacy. So an efficacy of 90% will require a serum dilution of 1 in 200. If we go down to a 1 in 32 dilution, we would only get a 50% efficacy. But you know, this obviously has important implications for thinking about well, other antibodies, but also about vaccines. So what are the implications of the AMP trial? Well, so as I mentioned, you know, that the trial overall did not show efficacy, and that's because fewer than 30% of circulating strains are that sensitive to VRC01. And so what that means is that we need more potent antibodies, and of course we need combinations to increase the efficacy. But what we also, as I've just shown, protection will likely require fairly high levels of serum antibodies, probably higher than we you know, would have expected from you know, from the non-human primate studies. But what AMP has also done is it has validated our in vitro assays. We now have a biomarker, and it also validates our NHP models. And that's really important in terms of thinking about the next steps, that we can rely on these in vitro and these experimental systems to design future trials. I think another important thing is that AMP showed us that antibody efficacy was independent of gender, population, clade, dose, or route of acquisition. So as part of when this paper was published, Bruce Walker wrote a very nice editorial called The AMP Trials, A Glass Half Full. And there has been, I guess, some people have been very optimistic about the results from AMP, and some people have been less optimistic. And I guess it does depend if you're a half glass full or half empty kind of person. I'm certainly the optimist, and I see then that we can fill the other half of the glass, because we need to remember that VRC01 was the first available product. It was first available antibody, apologies. It was never intended as a product. But it's given us the signal that we need to show that bNAbs work in humans. In fact, even if AMP had shown efficacy overall, I'm not sure we'd be in a different place to where we are now. It's the signal that we needed, and we have the signal. And we know now that we have much better bNAbs that are 10 to 50-fold better than VRC01. And so this signal is going to allow us to test these better bNAbs. And indeed, there is a robust pipeline of bNAbs ready to be tested for HIV prevention. They target different sites. So I've talked about CD4 binding site antibodies, and there's other ones as well in the pipeline. There's V3 glycan, there's V2 glycan, and there's also a 10E8 antibody. And then there's also these multi-specific antibodies, which are also very exciting, and we really await to see what the PK of those antibodies look like. But I do just want to highlight these two trials, a phase 1 trial of a triple combination, the Hvtn 130 HPTN 089. And then I'm also just quickly going to tell you about the Caprese trial, because it is a trial that I have been involved in. So this is the Caprese 256 antibody. It's a highly potent antibody that we isolated together with the VRC back in 2014 from donor Cap256 from KwaZulu-Natal in South Africa. It targets the V2 region of the HIV envelope. It's highly effective against clade C viruses. And in combination with Dan Baruch and Boris Yulg, we showed that it protects monkeys at very low dose, so that super potency in vitro translated to in vivo. The GMP lot has been manufactured, and the phase 1 safety and PK trial has been completed. But just to also, let you know that this is not smooth sailing, because this antibody is highly unusual. It's got a very long CDRH3, unusually long, and it in fact has a clipping site, or it's sensitive to proteases in the CHO cultures. And Nicole and her team were very carefully identified that this is due to a lysine, and so by changing it to alanine, they were able to prevent the clipping, to prevent the protease digestion. This had no impact on the neutralizing activity. However, it did delay the manufacture of the antibody by 18 months. So really just speaking, this is, antibody manufacture and production is, I guess, uncharted territory in some ways, and, lots of things in the way, but I'm very grateful that the VRC have helped us get around these obstacles. So in terms of the trial, which Slim Karim is leading, this trial started a year ago, and it's the first in human trial, and this is to test, so the V2 is this version with the clipping mutation, or to prevent the clipping, and then it's an LS version, and it's being tested alone and in combination with VRC07523LS. It's a dose escalation study, both IV and sub-Q, and the sub-Q is being enabled through the use of hyaluronidase, which basically temporarily disrupts hyaluronic acid that surrounds cells, and so it allows for a larger amount of antibody to be injected and absorbed. The results so far is that this antibody is safe and tolerable. There have been no major safety issues. The PK shows that it's very similar to other MAB, so nothing unusual, despite the fact that it is an unusual antibody. And the predicted modeling done by Edmund Caparelli shows that with that combination of CAP256 and VRC07 at 20 mg per kg, we'll be able to maintain serum concentrations of more than a mg per mil for up to 16 weeks. And the phase 2 trial has now been approved and is due to commence in Q3. So one of the things that we've also done is to use the data from the AMP trial to try and predict both the coverage and the efficacy of these combinations of antibodies. And actually here I'm just focusing on clade C viruses. We actually used the data from the AMP trial, so the AMP viruses. There were 47 in the placebo arm, and tested against all of these different antibodies. And as you can see, VRC01, as we know, only hits about 30% of viruses at this concentration at 1 microgram per mil. And certainly the other single antibodies you can see are much better than that. And then just focusing now on the triple combination of CAP256 with VRC07 and PGT121, that we get up to almost 90% coverage at this very stringent cutoff. And very similar results as well with the PGDM 1400, up to 90%. And in fact, even if it's, in fact, it's up to 50% if you're looking for dual activity, so two antibodies active in the mix. And so the PGDM 1400, VRC07 and PGT121 is in fact going into a phase 1 trial starting in September. And so what Yunda Huang and Peter Gilbert have done, have predicted the prevention efficacy of this triple combination following a single infusion, and looked at it over 16 weeks. And again, we're just focusing here on clade C, and just focusing on the from viruses from the 703 AMP trial. But what you can see in green at the bottom is the VRC01 data, and you can see that over time, the efficacy is, drops, and so there's no efficacy after 2 weeks. And if you look at the triple combinations, you can see it starts off with a very high level of efficacy, and after 16 weeks, we still have 80% efficacy for the dose of 20 mg per kg, and higher if we use 40 mg per kg. So really very impressive that we'd be able to maintain high levels of protection with this triple combination. So what does the future hold? Well, HIV still represents a formidable challenge and an urgent one. And I hope that we can learn a lot from the COVID experience, both in terms of just being faster about running trials and getting products into people. But what we have shown here is that the AMP trial has shown that bNAbs can prevent HIV infection. We have a triple bNAb combination that should show high levels of efficacy for 4 to 6 months. And so that really starts to become a feasible product if something only has to be used every 6 months. And passive immunization could have a major impact on HIV prevention, particularly in high-risk groups. And I certainly see that as being a targeted intervention. But what AMP has done is it's also provided us with a strong rationale for pursuing BNAB-inducing vaccines. And as I've shown, we have many new exciting BNAB concepts in phase 1 trials. So with that, I would just like to thank the fabulous team at NICD, and particularly to thank Penny Moore and her leadership over this time in taking the lab forward to new heights, and particularly also into the COVID world. But to thank all the other people in the lab, and also just to mention Nono, who helped run the AMP trial in our lab, but the whole team really just a fabulous. And then just to thank all of our fabulous collaborators, all long-term collaborators at Caprese, UCT, Duke, Hvtn, the VRC, and University of Washington. And then most importantly, to thank the AMP study participants. This was a landmark trial, and really to thank them as well for their participation. And with that, I thank you.